A pleasant morning to everyone, to my fellow classmates, and of course to you, Ma'am JP. In today's presentation, we are going to discuss all about autoclaves, its types, its practices, and of course its principles. Listed here are the content found in this presentation.
Presenting to you the objectives of our lesson. First is to define an autoclave and identify the different types. Second is to explain the mechanisms of how an autoclave functions. Third is to determine the basic thermodynamic principles that govern the operation of an autoclave.
Before we proceed, let me introduce first the reporters in today's discussion. First, we have Yrany Nicole, Ebron Makailas, and of course, yours truly, Nicole Padugdog.
Do you know what an autoclave is? It is a machine that uses steam under pressure to kill harmful bacteria, viruses, fungi, and spores on items that are placed inside a pressure vessel. It is also known as steam sterilizer. Autoclaves are commonly found in hospitals, laboratories, dental offices, and manufacturing industries. It is considered as more effective method of sterilization as it is based on moist heat sterilization, and they ensure safety and reliability by using high temperature and high pressure steam. Let's proceed to the uses of an autoclave. It is used for sterilization, regulation, curing composites, decontamination, and material testing. Medical instruments, surgical tools, laboratory glasswares, injectable medications, and components were sterilized using an autoclave. It is also used for regulation. It is a critical process for ensuring safety, quality, and compliance in various industries. By understanding the regulatory requirements, implementing proper quality control measures, and monitoring the performance of the autoclave, organizations can ensure the effectiveness of this sterilization method. Additionally, autoclaving is a highly effective method for decontaminating biohazardous waste, such as laboratory cultures, medical waste, biological samples, and contaminated equipment such as healthcare suits, safety glasses, and respirators. It is also important in material testing to test the durability of materials, and curing composites to enhance the properties of composite materials. Types of an autoclave. First is the pressure cooker type. It uses steam under pressure to kill microorganisms, typically around 121 to 134 degrees Celsius. Certain materials are metals, glass, and plastics. Second is the gravity displacement type autoclave N-type. Loads are sterilized by pumping steam into an ambient air autoclave chamber. The steam then displaces the ambient air by gravity. It needs longer sterilization and drying time, and at around 121 degrees Celsius. Third, positive pressure displacement type B-type. It utilizes an independent steam generator to create steam, often operating up to 134 degrees Celsius. It has a shorter total cycle time. It is better to use in hollow instruments and heat-sensitive items. And fourth, the negative pressure displacement type S-type. It contains both the steam generator and a vacuum generator. It has shorter time cycle and often operating up to 134 degrees Celsius. It is better to use in heat-sensitive items. Now let us discuss the parts of the gravity displacement type autoclave. The steam chamber is the main area inside the autoclave where items are placed for sterilization. The steam jacket. It is a place surrounding the chamber that fills with steam. Perforated shelf. It is a shelf with holes to allow steam to circulate around items for even sterilization. Door or locking mechanism seals the chamber to ensure effective sterilization. Next is the operating valve that controls the flow of steam from the steam jacket into the chamber. The safety valve that opens automatically to prevent explosions. The automatic ejector valve ejects air from the chamber, while the exhaust valve opens to release steam after the sterilization process. The pressure gauge. It displays the current pressure inside the autoclave. The pressure regulator manages the amount of steam entering the autoclave by controlling the pressure from the steam supply. The thermometer measures the temperature inside the steam chamber. And the path that provides the autoclave with steam is called a steam supply line. There are three different steam sources. First is the internal steam generator. Water is heated within the chamber itself to produce steam. Second is the external steam system. The steam generator is located outside the autoclave. Third is the centralized steam system, where multiple autoclaves share a single steam source. And the last part is the waste line. It is responsible for the safe disposal of condensate and any residual waste generated during the sterilization process.
Now, let's move on how it works. The functionality of an autoclave can be simply compared to a pressure cooker by utilizing steam to create a high-pressure environment and maintaining a temperature too high for microbial survival. Autoclaves are capable of successfully eliminating pathogens which may be resistant to soaps and detergents. The pervasiveness of the steam created within the autoclave allows a thorough sterilization within hollow instruments and other tiny vents which could be missed during cleaning with detergents. Let's discuss the four phases on how a gravity displacement autoclave functions, namely the steam injection phase. Next is the air displacement phase, then the sterilization phase, and lastly the exhaust phase. In the process of autoclave, the steam is needed. Before the steam injection phase, the steam is produced through boiling using an industrial boiler. Steam injection phase, the jacket phase. Steam is introduced to the steam jacket, ensuring that the chamber walls are heated up first in order for the steam to not condense later, preventing the items being sterilized to be wet. Steam injection phase, chamber phase. Steam is introduced into the autoclave chamber to use during sterilization phase. Air displacement phase. As the steam enters the chamber, the steam rises to the top due to its low density, displacing the air out through the drain vent and towards the waistline. So here's an illustration of the steam's mechanism. The red arrow indicates the hot steam as it enters the chamber. Once inside, the steam rises to the top of the chamber, displacing the cooler, denser air. The cooler air is forced out of the chamber and goes through a drain vent. As you can see from the illustration, there is a blue arrow and that indicates the cooler air being forced out of the chamber. Sterilization phase. As the appropriate temperature and pressure is achieved inside the chamber, and as a conduction occurs between the steam and the materials, the microorganisms are killed. The steam supply valve is shut off when the pressure inside the main steam chamber reaches the target temperature and pressure. In modern autoclaves, this is electronically shut off. In the graph, the temperature and pressure must be maintained during this time period in order to properly sanitize the items in the autoclave. In modern autoclaves, this is monitored automatically and begins the cool-down process accordingly. During the cool-down phase, the pressure in the steam chamber must be maintained until the temperature drops below 100 degrees Celsius to prevent containers with liquids from boiling over and spilling into the autoclave. In this example, the exhaust valve is gradually opened while closely monitoring the pressure gauge to ensure the proper pressure in the chamber is maintained until the temperature inside falls down below 100 degrees Celsius mark. The ideal temperature and pressure for an autoclave to sterilize equipment and supplies is at 121 degrees Celsius or 250 degrees in Fahrenheit and 15 pounds per square inch of steam pressure. Exhaust phase. Once the sterilization cycle is complete, the exhaust valve is gradually opened to release the steam and the lower pressure and temperature inside in order to cool down the chamber and the materials that are sterilized.
After knowing the process, this time, let us understand the way an autoclave works. There are three variables affecting an autoclave: temperature, pressure, and time. As temperature increases, the pressure increases as well, and as temperature increases, the lesser time it takes for sterilization. Going back to the process, as water is heated to produce steam, there is a transition from one phase to another known as phase change, and the required energy needed to transition from liquid to vapor without an increase in temperature as it reaches its boiling point is known as latent heat or latent heat of vaporization. When steam enters the chamber, three types of heat transfer is observed. As steam engulfs the jacket and the chamber along with the materials being sterilized, conduction is observed, where heat slowly penetrates the outer surface exposed to the heat and to the next layer until the entire material reaches the proper temperature for sterilization. As the steam circulates to distribute the heat, it creates convection current that helps maintain consistent temperature and pressure. Afterwards, as the autoclave operates to sterilize the items inside, some heat may be lost through infrared radiation from the hot surface. It was also previously mentioned that the autoclave is affected by three variables, and two of those is pressure and temperature. In gas law, under Gay-Lussac's law, it states that the pressure exerted by a gas varies directly with the absolute temperature of the gas. This is observed as temperature increases due to steam. The pressure inside the chamber rises accordingly. The temperature kills the bacteria, while the pressure is responsible to rapidly spread the heat within the materials. Apart from the previously mentioned principles, the first law of thermodynamics is also observed in how an autoclave works. This is the interaction between heat, work, and internal energy. An autoclave is a closed system since only energy is exchanged in the form of heat. Inside the autoclave, both heat transfer and work are involved. Heat enters the system, which in this case is the chamber and the materials inside it, in the form of steam at high pressure. The steam transfers heat to the materials, raising the temperature to the required sterilization level. And since the autoclave operates under high pressure, the steam inside the chamber does work on the walls and the contents inside the chamber as it expands and compresses. During sterilization phase, the internal energy, heat, and work increases as both the temperature and pressure increases. However, during cooling phase, since the temperature and pressure decreases at this point, the internal energy, heat, and work decreases. Furthermore, the second law of thermodynamics, or also known as entropy, is also apparent. Entropy is the measurement of disorder within a system. The increase in temperature and pressure contributes to the increase of entropy inside the chamber. As the steam penetrates the materials being sterilized, it transfers heat, increasing the system's entropy due to the presence of heat in the material and the increase in kinetic energy of the particles. As temperature and pressure increases, the kinetic energy of the molecules also increases, increasing the entropy of the system. That ends our presentation. Thank you so much for listening, and if you have any questions, feel free to ask them and we will answer them to the best of our abilities. That would be all. Thank you.